Greetings, colleagues and friends, and welcome to this, the first online FameLab South Africa final. And we are very excited to be sharing such wonderful work by incredible scientists and, of course, incredible science communicators at this event. We've been running the FameLab competition around South Africa. We are grateful to the partners who have hosted us all over South Africa. We have developed fantastic relationships, and we have grown an awareness and appreciation for the importance of science communication, the importance of research which engages with and for the society in which it operates. And that's really the purpose of this FameLab competition. FameLab was started in 2005 at the Cheltenham Science Festival. Since then, it has spread to over 30 countries around the world. South Africa has run the competition since 2013, and we do that in South Africa in collaboration with, or in a partnership between ourselves, Jive Media Africa, a science communications agency, the British Council, and of course the South African Agency for Science and Technology Advancement, which is a business unit of the National Research Foundation. The FameLab competition exists to grow talent and to identify talent for science communication. So it's not enough for scientists to just do science. We really need to find ways in which scientists can talk to the public. And we're really excited. We've traveled around South Africa and partnered with a large number of organizations. And at each of those spaces, we've run training in science communication, in public speaking, and then we've held a final. At each of those institutions, the finalist has been selected out of between 10 and 20 or 30 participants. And those finalists got gathered together for a 4-day masterclass training with a UK-based trainer. That was Jamie Gallagher, who was brought to the competition in partnership with the British Council. And those participants gained insight not only in how to do public speaking, but in fact, due to this new space in which we're operating on an online space, a lot of input about how to communicate their science to public audiences through the medium of video conferencing or meetings online through digital means. So we're very excited to share this competition with you. We believe strongly that an empowered society is one that is able to identify problems, investigate those problems, and apply new solutions with a critical reflection on how those solutions are going to impact our futures. And we believe strongly that the FameLab is the space in which that real passion and commitment to finding those solutions and sharing those solutions with public audiences can take place. So from my side, thanks very much for being part of this. Thanks for watching. And we're looking forward to growing the support for the FameLab initiative. Most of all, congratulations to every single one of the participants who you will be seeing today. They've done an incredible job. They're growing in leaps and bounds as science communicators, and we're looking forward to exciting developments in this space. Enjoy.
Right, judges, I'm going to present to you our first participant for this, and that is Dr. Nabila Ismail. She has successfully completed her PhD studies. Furthermore, Nabila aims to reduce negativity and inaccurate information surrounding infectious diseases by communicating vital scientific facts to the general public in a way that can relate and understand. Nabila, if you are ready, we are standing by.
Good morning. My name is Nabila Ismail. I want to take you back 100 years ago when a scientist returning from a vacation found that fungus had contaminated plates on which he was growing bacteria. And the fungus appeared to be releasing a substance which was inhibiting the growth of those bacteria. This was the discovery of penicillin, the world's first antibiotic, and it was a discovery that changed the face of medicine. 20 years later, another antibiotic was found to be effective against the TB bacteria, or the bacteria that caused tuberculosis, a disease which at that time was considered fatal. Now let's fast forward to today. At a time when it is known that TB is the leading cause of death due to an infectious agent. But how is this possible, given that streptomycin was thought to be the cure all those years ago? Well, it's due to a phenomenon known as drug resistance. And I can imagine that if those drug-resistant bacteria could speak, they would probably sound something like this. I grew strong, I learned how to carry on, I will survive. Because as long as TB bacteria know how to mutate, they know they'll stay alive. Mutation, or the change to the genetic code inside a cell, results in physical changes which renders a drug ineffective. How is this possible? Well, a drug can bind to its target inside a cell, making it effective, similar to two jigsaw puzzle pieces being compatible. But when a genetic change occurs, it physically alters the target of that drug, rendering the drug ineffective. My research focuses on identifying those genetic changes which prevent drugs from being effective. Through using sequencing instruments, we can identify genetic changes to find which drugs will be effective for a patient and which ones won't be. We can then prescribe a combination of 4 to 5 drugs. This was not done for streptomycin all those years ago. Through reading this genetic code, we can then prescribe effective regimens with combination therapy. This will prevent the spread of drug-resistant tuberculosis and protect the limited number of drugs we have available for the treatment of TB. Thank you.
Right. Thank you very much, Natasha. Your time is now up. All right, Natasha, let's start with you.
Hello, Nabila. Well done. It must be terrifying to speak into the void when you're used to having people kind of engage back with you, but you did really well. I thought you paced yourself really nicely, and I could see that you were nervous, but you kind of didn't trip over the mistakes that you made, and you have a very nice, warm, kind of natural presentation style. So well done for talking to all of us imaginary people. We are real, I promise. I think the work you're doing is really important. What I'd be interested to know is how long would a process like this take of this kind of sequencing, and how long before it would become something that could kind of come to market, I guess?
I think that's the main focus of my research, is to reduce that time to result. So we're working on every aspect of sequencing directly from a patient sample. And currently, if you use the conventional methods, it can take months to actually get that result, because then you have to grow up the bacteria and actually culture them and then extract DNA. But we're trying to extract DNA directly from a patient's sputum. We want to do this within 2 weeks, and our first pilot studies have shown that we can actually get it done, but it's very, it's highly contaminated, so we're struggling a bit.
Back to the drawing board a little bit.
Yes.
Awesome. Well, thank you very much, and good luck with the rest of it. I'm going to hand over to whichever judge is speaking next. Sorry, I've lost track. Nasipo, I think.
Hi, Nabila. Thank you so much for such an engaging talk. I love how you light up when you speak about TB and how you still maintain eye contact and try to use props very much effectively to enhance your talk. I think that was very brilliant. It was very engaging. It made it easy to follow through what you were talking about. My question for you this morning would be, with tuberculosis having been in existence since way back when, why do you think it is still important till this day to still do research on TB?
So I think when we look at what's available for TB research, the fact that the disease is curable, it's actually sad that so many people are dying from this disease. I mean, 1.5 million people died in the past year alone. And that for me is actually tragic, that a disease that's curable and preventable and treatable still continues to play such a big role in our society. So that's why I think the research is more and more important. For example, if we just took TB as half as serious as we're taking this coronavirus pandemic, we could have radically reduced those numbers. And I find it really sad that those measures weren't put into place. And of course, there's a whole lot of politics around it. But I wonder if it wasn't a third world country problem, and maybe if it was affecting the first world countries like cancer and diabetes, would it be taken more seriously then?
Oh, thank you very much, and good luck.
Thank you, Nosifo.
All right, thank you very much, Nabila. That'll be all from your side.
Thank you, Star.
Next participant is Johannes Huber. As part of his MSc in zoology at FABI, Johannes Huber studied how differences in the chemical composition of leaves of different eucalyptus species could alter the beetles' feeding behavior. This led him to his PhD now in zoology, which focuses on the relationship between the beetles, eucalyptus trees, and the parasitoid wasp. Johannes, the floor is yours.
Thank you. Good morning, everyone. I want you to imagine that this morning, when you woke up, you tried to move your legs, but they wouldn't budge. So you tried raising your arms, but they hung at your side. Okay, you tried opening your eyes, but no, they remained sealed. You were in a world of absolute darkness, and you couldn't move a muscle. And then you feel it. Something is crawling up your leg, and all of a sudden, it bites down. Now, for us, this is a nightmare. But for a tree, this is just its normal life. Fortunately, however, trees are a bit better adapted to this nightmare than we are. You see, when one of these monstrous insects crawls up the tree and takes a bite, well, the tree has these interesting defensive compounds that float around in their tissue. And these can do anything from tasting foul to straight up killing that insect. So trees, they're well defended. But there is a creature that doesn't really care. And the tree is as vulnerable to it as we would have been in the tree's position. What kind of monster is this? Well, it's this, a eucalyptus snout beetle. Just a tiny beetle. Doesn't seem very dangerous. But you see, when you can't move, when you can't fight back, even a tiny beetle can become a nightmare. And this is a real nightmare. You see, a few of these beetles get into a plantation, and then they start multiplying. And soon you'll see swarms of them stripping the trees of their leaves. And without leaves, the trees don't grow. And all of us that are so dependent on those trees, you know, for the wood we use, for the paper we write on, even for the air we breathe, all of these benefits get taken from us. We're forced to sit back and watch as these woody friends of ours are torn to pieces. And I won't stand for it. I will defend these trees and I will fight this monster. But how do we fight? Well, we have to use science. So I started by studying the system. I tried to understand it. And then I noticed some of the trees weren't as injured as others. But why? So I looked into it. And then I made my big discovery. There were 5 compounds that actually did affect the beetle. But because in nature they're either at the wrong concentration or in the wrong ratios, the effect is mild. But we know what we have to know now. If the concentrations are wrong, we just have to make a new tree, one with the right levels. And then the tree will defend itself, and we can all sit back and enjoy the benefits that come with having a healthy and happy tree. So thank you for listening to me. Do you have any questions?
Thank you very much, Johannes. Okay, Nozipho, shall we start with you? Nozipho Gumbi?
It's Prof. Knox turns.
Oh, sorry, Prof. Knox? This one is yours.
Wonderful, lovely presentation, Johannes. I really enjoyed it. And I really want to commend you on how you were modulating your voice and using your body. I thought that was really positive. It kept us engaged. And I like the way you're quite animated. Again, it really is very engaging. So really well done. And the use of really simple terms to try and explain some really complicated chemistry as well was absolutely wonderful. My question to you this morning, though, is how would you defend the work that you are working on eucalyptus when eucalyptus actually happens to be an invasive species?
That is a great question. Now, eucalyptus is a problem. Some eucalyptus, though. There is invasive species that cause a lot of harm. But eucalyptus is an incredibly diverse genus. You know, it's like there's a lot of different trees and they do a lot of things. Now, some of them bad, but a lot of them are very fast growing and they don't really cause a lot of harm. So when we plant these in plantations and, you know, mass rear them, we can get a lot of wood that's quite good. We can use this for our papers, for general timber, mining timber, and these are a backbone of the economy. It's one of the, I think it's either the largest or second largest wood producing plant we have. So it's a major part of our economy, and without it, this country would be far worse than it is now. So we need eucalyptus and we need to protect it. So that would be my answer to you. Thank you for the question.
Great, thank you.
All right, shall we take our last and final judge?
I think that's me. Hello, Johannes. Well done. I thought that was a very engaging presentation. I'm going to have thrills for the rest of the day, imagining something crawling up my legs while I'm sleeping. But it was a lovely use of a metaphor. I think that sometimes metaphors can be quite badly used by scientists, but you used that one really well and you carried it through really well. What I want to know, given that I don't have Prof. Mukunga's deep scientific background on this kind of stuff, what does it entail to make a new tree? What's the kind of process of the genetic work that you need to do?
All right, so there's two approaches. There's the very basic one where you look at a tree, you say this one has compound 1 really high, this one has compound 2 really high, you just cross them, now you get one with hopefully one of the children has both compounds high, just very basic breeding. Then there's the other approach, which is more technical. You have to sort of figure out what genes cause the compounds, then you screen the trees for those genes. And then you can say, okay, we have gene X, G, Y. If we cross them, we'll get this hybrid, and this will produce X amount of. So it is a lot more work, but generally it's the better approach because you can get a more tailor-made tree. So effectively, it's a lot of general screening. You'll have to look at hundreds of trees, say, what's here, what causes what's here, and can we use it? If that answers your question.
It does. Thank you very much, and good luck.
Thank you very much.
All righty. Thank you very much, Johannes. That will be all for now. Thank you for your time. All right, judges, up next we have Josephine Kifelek. A single wasp is a mild nuisance to deal with, so imagine the damage an entire swarm can cause. Josephine Kifelek, who has specialized in the ecology and the evolution of insects, now focuses her PhD research on combating the damage caused by these invasive insects. She believes that conveying this passion to the public will reduce misinformation and help raise awareness about such issues by explaining precisely why everyone ought to care. Josephine, the floor is yours.
So did you know that the deadliest animal on the planet is actually a mosquito? Well, diseases carried by mosquitoes, such as malaria, dengue fever, cause every year 1 million victims. And 90% of these victims are in Africa. So what if I told you that to fight this battle, we could harness the power of the gods? But not any god. Cupid, the god of love. In the insect world, Cupid does not look like a cute cherub with a bow and arrow. He rather looks like this, a blob. In fact, our Cupid is a bacteria. His scientific name is Wolbachia, and he lives comfortably in the reproductive tissues of mosquitoes, bees, beetles, most insects actually. And by being present in their reproductive tissues, Cupid is able to become nature's most ruthless matchmaker. In fact, these bacteria, just like humans, can come from different families and have relatives. So for the mosquito, this Cupid has one rule. You may choose any partner you want, as long as they carry a bacteria that comes from my family. So, when a male mosquito and a female mosquito choose a partner that carry the same bacteria, Cupid will allow them to form a pair and to reproduce. Because they followed his rule, Cupid will even gift their children with superpowers. This can be resistance to parasites or a very efficient digestive system. However, if a male mosquito and a female mosquito dare to defy Cupid and pick a partner that carries a bacteria that is from a different family, they will be punished. In fact, using a toxin, Cupid will kill all of their children while they're still embryos. So how can we, as scientists, harness this power and fight against diseases carried by mosquitoes? Well, in the lab, we can create armies of millions of these male mosquitoes. We then inject them with a bacteria. But this bacteria has to be different from the one in the females in the wild. We then release those males. And because male mosquitoes do not bite, they are not going to be a threat to humans. They would rather spend their time finding and courting females trying to reproduce. But because they carry different bacteria, they will not be able to reproduce. They will not be able to produce the next generation of deadly mosquitoes. So while they make love, we make war. Thank you.
All right, thank you very much, Josephine. Okay, Nozipo, this one is yours.
Hi, Josephine, and well done on giving such an engaging talk. I love how you opened up with the question, posing the question to the public so as to captivate and capture our attention. I also loved how effectively you used your props and the metaphor behind Cupid bringing together the male and the female mosquito. I also learned something new, that male mosquitoes do not bite. So thank you very much for that. So my question to you this morning would be, what other interesting facts would you like to share with the public about mosquitoes? Because as far as we know, mosquitoes bite and we just don't want them anywhere near us. But is there anything else that's interesting that you want the public to know about mosquitoes and the research that you do?
So one question that I get very often from my friends that don't know anything about insects is that, why are they mosquitoes anyway? What are their roles? Is it just to kill things and to suck blood? Well, actually, as I told you, male mosquitoes don't bite, but they have a role in our ecosystems and in our environment. And a lot of them are actually pollinators for plants and flowers. So they are really important to keep our trees and flowers alive. And they also are a food source for like frogs and fish. So they're also very important to keep these populations alive. So mosquitoes are not just there to suck blood, they are also there to maintain, and they have a role in our environment.
Thank you very much. That was lovely. Good luck.
Welcome. Thank you.
Great. I have some comments as well, and I really also loved the way that you used your props. I thought that was really wonderful. Great drawing skills. So that must have been quite fun to make as well. And I like the way that you were very engaging and use your voice and great eye contact. It was very interesting. I wanted to better understand this morning if there are other biocontrol mechanisms that would be able to control malaria, or is this the only system that we really have available to us?
Okay, so I could give two answers to that. You can also, so using that army of male mosquitoes, you can also use radiation to make them infertile. So they will take the place of male mosquitoes that are not fertile, and then try to fertilize females and fail. And another way you can actually use that bacteria that I'm talking about is that some of these bacteria have the capacity to be in competition within the mosquito with other bacteria that could cause disease. And so by putting these good bacteria inside the mosquitoes, they will actually prevent bad bacteria from living within the mosquito.
Excellent, thank you.
Thank you.
All right, that'll be all for now. Thank you so much, Josephine.
Thank you very much for everyone for listening. Have a good day.
All right. Thank you. Judges, up next, we have Lerato Ndlovu. Lerato has completed a master's degree in biochemistry and plant breeding, is currently pursuing her PhD, and chose to focus her research on TB immunology in order to improve TB cure rates. Lerato, the floor is yours.
Thank you. Hi, I'm Lerato. Over the past 50 years, scientists have learned how to read and write the human genome. We've developed birth control pills. We even have robots that can perform surgical procedures doctors spend years learning. But what we haven't been able to achieve is the end of TB, a bacterial lung infection that kills 5,000 people every day. Nor have we been able to develop medicines that can shorten treatment time, despite the fact that getting people to take their treatment for the prescribed period is a challenge in controlling the spread of infection. But to develop these drugs that can shorten treatment, we need ways of knowing, consistent ways of knowing when a person has been successfully cured. Sputum culture is the gold standard in determining how well or how poorly a person is performing on treatment. It involves coughing up mucus from your lungs that can be tested for the presence of bacteria. It is also the method used to diagnose TB in resource-limited settings, but it's not very sensitive, meaning that people who are unable to produce this mucus can sometimes go undiagnosed, such as children and those living with HIV. My work uses blood to study the immune system response to an infection, to a TB infection, in order to find a simple and sensitive way of knowing when a person has been cured. The immune system functions much like an army. It has lots of different cells with lots of different functions. Some are to kill and destroy invading bacteria such as TB. If this were your immune system, neutrophils would make 60% of the soldiers. These cells are often the first to arrive and the first to engage the enemy. They also have a very short lifespan of about 2 days. These cells are able to release chemical signals or messages that attract other cells to the site of infection or the battlefield. Not only that, but they can also release chemical signals that tell the army that the fight is over and the enemy is destroyed. Using just 10 drops of blood, I am able to study these neutrophils by labeling with fluorescent colors that when passed through a flow cytometer just a big laser machine, I'm able to determine the brightness or the dimness of these cells in order to understand how they are associated with treatment response. I can monitor this brightness in order to find a specific and sensitive signal that can correspond to treatment success. And I'm proud to announce that as of yesterday, the work from our study has been accepted for publication, because together we can beat TB. Thank you.
All right, just on time, Lerato. Well done. All right, shall we take Natasha for this one first?
I'm just waiting for my camera. Hello, Lerato.
Hi.
Must be very weird to talk into nothingness, but what an amazing speaker you are. I could listen to you talk for hours, whether it's about TB or something else. Your body language is excellent. The way that you modulate your voice is great. The excitement that you feel about your research is, well, forgive the pun, it's contagious. So well done. That really fired me up and left me feeling super interested. So I thought you explained the science really well, considering that I have absolutely no scientific background. So I wanted to ask you, given your different kind of background at a master's level, what was it that drew you specifically to the immunology of tuberculosis now that you're at PhD level?
So I've always wanted to work on something that I thought was important. And in the South African setting, there's nothing that's more important currently than TB or HIV. And so when I had an opportunity to transfer the skills that I'd learned in my master's to this field of immunology, I just jumped at the opportunity. And yeah, that's it.
Awesome. Thank you so much and good luck. I'm looking forward to reading your research when it comes out. Good luck for this competition.
Thank you.
Good morning. Oh, yeah, still morning. Good morning. That was really wonderful. I thoroughly enjoyed it as well. I loved your passion. And I really also loved the fact that you started us off with some, a fact that was a really good start, because it really got us engaged and connected to the subject. I love the fact that you coughed in the middle of your talk. I think all of us are quite engaged with respect to coughing, so that would be a really one for making people to pay more attention. And I also love the fact that you took ownership of your research when you said, what you were doing, when you started to talk about what you were doing, and you said I think that's a good thing as well. I just have a very small question. So the paper that you've published, what's it on? Is it on this topic or on something else?
It's on this topic. So as I mentioned, we're trying to find a marker or a signature that can be used to determine quite early on if a person has been successfully cured of treatment of TB. And the whole point of it is to be able to shorten TB treatment for those who have trouble complying with the long 6 months of treatment. That's 480 tablets. And so, yes, so this is the work that we've published. We managed to find a signature, and soon when the paper's out, I'll be putting it all over social media. So watch out for that.
Great, thank you.
Thank you.
All right, thank you very much, Lerato. That will be all for now.
Okay.
Have a
Thank
lovely
you.
afternoon.
Bye. You too.
Bye. Judges, our next participant is Sendibitiosi Gandizwana. He completed his master's degree in chemistry and is currently conducting his PhD research on a palladium-based drug to be used in the treatment of cancer. Sandy, the floor is now yours.
Thank you. Have you ever wondered, when will some of our global challenges end? Let's think about the money spent on weapons or the time and resources spent on anti-corruption inquiries, or the lives lost during wars, be it the soldiers, our heroes, or civilians, just collateral damage? Can I trust you with a secret on how to stop or reduce this? When I was 5, my mom told me that inside our bodies we have soldiers, and they fight enemies. Sometimes they get defeated or find themselves in compromised, tricky situations, and we get sick. Imagine if you were a soldier and you discovered that the enemy is your family, friend, or relative. Would you pull the trigger easily? Our human bodies are made up of normal cells, the good guys, law-abiding citizens. Sometimes they can turn into bad guys, cancer cells, terrorists. They become greedy, they multiply non-stop, they don't want to die, they want to use a lot of resources, and they want to spread everywhere. The good news is we don't negotiate with terrorists. So we are developing undercover special forces, drugs, that when deployed inside our bodies, their mission is simple: to identify and to kill the bad guys. The good guys can't see them, but the bad guys can see them. Bad guys think, wow, we just got new corrupt friends in the hood. They have the resources that we need. Undercover boys go and say, here we are. Bad guys say, wow, no, come in, let's dine together. They take inside. After or during dinner, boom, job done. Special Force 007 James Bond, thank you for dealing with the bad guys. You are free to leave our bodies. See you next time. This is specific and selective targeting of cancer cells through nanomedicine. This way, we overcome the current global challenges in cancer treatment. We save money, time and resources, and importantly, more lives. This was my secret. I hope it's safe with you. Please don't tell anyone. Nanomedicine changing the world. Thank you.
All right. Thank you very much, Sandy. Shall we then start with you?
Yes, thank you very much, Sandy, for such an engaging talk. I love how you came already prepared and dressed to address us with your lab coats and gloves. I love also your use of props to enhance the talk and enhance the message that you are trying to get across about the use of nanomedicine in dealing with cancer. And so my question for you this afternoon would be, a lot of times when we hear about these new strategies that are put forward for dealing with cancer, there's always a setback. While they do good in terms of treating the cancer, but there's also damage to some of the healthy or the living cells in the body. So in your particular work, how are you able to ensure that the good does not, there's a balance between the good and the bad, or that there's no bad at all that is happening?
Okay, thank you. So what we are doing is we realize that there are some differences between the cancer cells and our normal cells. So specificity and selectivity is one of our key components. So in so doing, we are targeting one of the receptors, a binding site that is mainly found only in cancer cells and not found in normal cells. So by so doing, we minimize, there's no intricate uptake that has been recorded up to now from our studies. So our normal cells don't actually take up our drugs. So our drugs are specifically delivered in there. One of the challenges is usually that delivery aspect. And another one is also of making sure that at least the drug gets out of the body. So in this case as well, we've also ensured that our biodistribution and clearance is spot on. So we have ensured that part.
Thank you very much. That was very good. And good luck for the rest of the competition.
Thank you.
Hi, Cindy, that was fantastic. You're a very engaging speaker and you're a man with a sense of humor, which I always appreciate because even serious science can be explained in a very sort of fun, engaging way. Your props are brilliant. I wanted to know what other areas of research nanomedicine is being used in. I know that it's not just in cancer treatment. Are there other kind of areas where researchers are exploring how nanomedicine can change the way we do medicine?
Thanks, Natasha. Yes, quite a lot of people sometimes they're also looking into nanomedicine is also being used and being applied in mainly drug delivery, whether be it in terms of viruses, in targeting viruses, or it can also be used in well, like for instance, right now there are a lot of other research that's trying to find in antimicrobial activities of the nanoparticles. So I'd say that nanomedicine is nanoparticles generally they are, for my kind of interest, it's mainly for the nanomedicine aspect, but it's being used in cancer. It can also be used in some people are using it to deliver some things in diabetes, and some people sometimes can also even target actually the mind to deliver some specific things in the mind. So there's quite a lot that people can be able to do, and they can use it mainly in drug delivery.
Fantastic. Thank you so much, and good luck for the race.
Thank you.
Judges, our next participant is Dr. Brenda Degama. Her participation in FameLab has equipped her with the skills to effectively communicate her scientific research to a broader audience and has given her the confidence to continue in her campaign to educate the public about how body donation contributes to science. Here's to contributing science. Brenda, the floor is yours.
Hello, I'm Brenda Degama. Extra, extra, come hear all about it. Come hear about what, Brenda? Come hear about body donation. Body donation is the giving of one's dead body to science. We use the donated bodies to teach human anatomy to health professionals. Now, health professionals could be the medical doctor that you may see when you are feeling sick, or the nurse that may assist you in hospital when you would have gone for a small procedure. Now, we use these bodies, and that's where I come in as an anatomist. We use them to teach health professionals about human anatomy and the different organs that form the human body, such as the liver, for instance, and the different lobes that it has. Or my favorite one, the heart. And we tell them about the different chambers, the different arteries that supply it. Now, what we need, as I've stated, are donated bodies. And when one day I went inside our lab, I realized and asked myself, but how come we are not receiving donated bodies from the Black African population group? You see, we receive from the white citizens, we receive from some Indian of the Indian population, and some from the colored population, but none from the Black African population. This prompted my research to find out what are the reasons behind the Black African population not donating their bodies to science. And this is what I found, that cultural beliefs were the reasons why they were not willing to donate their bodies to science. Now, when they don't donate their bodies to science, it means we are running short of one part of our rainbow nation, which is South Africa. And we need to know the different parts of the Black African population as well. Now, I'd like people, especially the Black African population group, to also consider donating their bodies to science. The reason is that, as the late President Nelson Mandela said, education is the most important weapon that we can use to change the world. And I'd like the Black African population to consider donating their bodies to science so that we can Heal the world and make it a better place for you and for me and the entire human race. Thank you.
All right. Thank you very much, Brenda, especially for that idol singing there in the end. All right, Prof. let's start with you.
Okay, thank you, Brenda. What a beautiful voice.
Thank you.
Wonderful, and I thought that was it was a really nice end because it rounded off your talk, which is actually quite sensitive, I think, for a lot of people. And I loved your use of props. I loved the way you used your body. And I think at moments where you needed to be a bit more sensitive, you toned down your voice, which I think was very good. I have one question. So you mentioned that it was cultural beliefs that is preventing the Black African population to donate their bodies. Which cultural beliefs are preventing people from donating?
Thank you for your question. The cultural belief that's affecting them donating their bodies to science is the belief in ancestors. You see, the Black African population believes that even though we as scientists think that once the heart stops working and the person has died, that's the end of life. The Black African population believes that you live and there's an afterlife. They also believe that while the living are still there, they need to connect with those who have passed on, and they have an influence into their living lives. So that is why they believe that they need to still have that connection with those who have passed on so that they can hear their wisdom and have guidance with their everyday lives.
Thank you so much for your answer.
Thank you.
I think it's me. Hello. Sorry, unless I'm accidentally stepping on Nisipo's toes, but I don't think I am. Brenda, that was wonderful.
Thank you.
I have a particular interest in body donation. I did some articles about it recently, and I thought that you added to my understanding. You're a very engaging speaker. I think it's quite nice to have a scientist who can sing as well. I think that's underrated. And yeah, I'd just like to echo Prof. Mukuga's comments. I thought that you kind of managed the light and the dark of it really nicely, because it is a very sensitive topic. And I thought your answer on the kind of cultural beliefs was very engaging. I'm curious to know, because of course it's not just in Black South African cultures, but in various cultures around the world, beliefs around ancestors and other kind of understandings around the body versus the soul. Do you know of work that's been done anywhere else in the world that perhaps we here in South Africa could draw from?
Yes, I have. Actually, I've got an opportunity in 2018 to visit New Zealand, which was a specific interest for me to learn about the Maori people, who also have similar beliefs like the Black African population as far as the afterlife. And I was very fascinating to actually see that in the anatomists there in the University of Otago have actually incorporated the cultural beliefs of the Maori, where they do a cleansing ceremony. Even in their dedication ceremonies, they incorporate like the wakotea and the clearing of the way for the spirit. So for me, it was a good learning experience where I could see how they're interfacing with the cultural beliefs of their population and still ensuring that the population donates their body to science. And of course, there are many other cultures as well, and some which still have strong beliefs as far as the afterlife, but and their anatomists have also used awareness to increase knowledge about body donation so that people can start considering donating their bodies, which is what I'm also trying to do in our country. So
Thank you.
Thank you so much.
That's really, I'm going to go and read up now on Maori burial cultures. So thank you very much and good luck for the rest of it.
Thank you so much.
All right, thank you very much, Brenda. That'll be all for now.
Thank you. Bye, Stabile.
All right,
Thank you,
I see that our next participant is. All right, judges, next we have Pinky Mukwena, a Master of Science candidate at the University of South Africa, a passion for environmental sustainability, together with communication skills honed by the FameLab competition, will enable her to enhance the lives of ordinary citizens while preserving and protecting the environment. Pinky, the floor is yours.
Awesome. So a friend of mine's grandparents recently celebrated 61 years of marriage, and I could not help but marvel at the type of bond that connects two individuals in this way. It wasn't long until I remembered that I know of another type of bond that exists between two individual atoms, carbon and fluorine, resulting in man-made organic compounds known as perfluoroalkyl substances, acronymed PFAS because of their obvious tongue-twisting shenanigans. These chemicals are popular for their advantageous use in industries for the production of furniture, insecticides, stain repellents, firefighting film foams, and recently, food packaging containers such as our takeaway boxes. Of course, if these essentials were the only offspring from this relationship, we would be popping bubbly and celebrating. However, because of the nature of their bond, they are non-degradable, which means that they can exist in our environments for years without ever breaking down. And worse, they can leach into our water systems, blending into the matrix, also known as polluting it. On top of that, they are infamous for their tendency to biomagnify, that is, find strength in numbers in our water systems, resulting in acute toxins that could cause cancer, among other known health hazards to humans. Moreover, their resilience makes them resistant to treatment through our conventional treatment techniques, which means that the need for affordable, easy to operate, and sustainable techniques of treatment has never been more vital. My research introduces the use of maize tassels. These are naturally occurring materials that are found at the top of male maize plant. They help with the fertilization of the plant, but once the cobs have been harvested, maize tassel is often discarded as a type of organic waste. Why maize tassel, and how practical is all of this, Pinky? I am glad you asked. When ground to powder under optimized conditions, maize tassel is able to chemically react with the functional groups that are found within these compounds, and as a result, nullify their potency. It's more like an internal makeover. With this, we get to feed two birds with one stone, and that is water treatment on the one side and organic waste management on the other. It's something I refer to as one of those instances where the answers to our complex, extraordinary questions can be found in ordinary places. Hashtag amaze. Totally amazing. Thank you.
All right, thank you very much, Pinky. Let's lead Nozipo.
Hi, Pinky, and thank you so much for such an engaging talk. It was such a pleasure to watch you and listen to you delivering your talk. I love how you use your body, the facial expressions to bring your points across, and the different variations in your tone. That was very impeccable. You mentioned in your talk that these maize tassels have got the potential to nullify the potency of these persistent pollutants. Do you care to engage with us a little bit more as to how the nullifying of the pollutants takes place, so that we are all clear as to the actual process by which this happens?
Absolutely. So Nozipo, there's a process called adsorption. Adsorption in general is when a material surface is able to attract the chemical makeup of certain objects depending on their ionic charges. So maize tassel is negatively charged, and it turned out that our pollutants are positively charged. So maize tassel is able to attract these negative charges into itself. As a result, it splits the connection, the bond, and that's how it does this. Yeah, so it's just because of its properties and attributes that it turns to be a viable solution to that.
Thank you very much, and good luck for the rest of the competition.
Thank you.
I just want to make sure that it's supposed to be me. Yes?
Yep.
Okay, thank you. Pinky, lovely to meet you, and thank you so much for a really great talk. I love the way that orange is actually popping on you. That's very, very cool on camera. And your voice was popping as well, and your tone was really lovely. I liked the use of props, and you know, you kept it quite simple and didn't use too many props, which I think was also very useful, because it meant that we kept our attention actually on you. I have a quick question about whether it's just maize tassel, or are there other plant species as well that could also be potentially utilized? Because we generate a lot of agricultural waste, and I'm just wondering if there are other opportunities to exploit this technology with other species.
At the moment, I only know of one research that made use of grape leaf litter. I'm actually planning to try to find another one through Moringa oleifera. It has the same properties as maize tassel. So for my PhD, I'm actually looking towards exploring that avenue, because I think it's another untapped resource that we could go into. So yeah, I'm pretty sure there's many others, but not yet discovered. So yet to be discovered.
Great, thank you.
Awesome, thank you.
All right, thank you very much, Pinky. Judges, lucky number 8, our final participant for this afternoon is Zakia Kasim, who is currently pursuing her master's degree in pharmaceutics and pharmaceutical microbiology at WITS, Advanced Drug Delivery Platform. Zakia believes that scientists ought to present their research directly to the public, as this research presents solutions to problems society is facing. This communication may also inspire younger generations to take up careers in science and further contribute innovation research to the scientific field. Zakia, the floor is now yours.
Ladies and gentlemen, why are you so calm when this house is currently on fire? This fire is burning in 75% of females' vaginas globally. This fire is not easily extinguished, as it is reignited every so often. This is a burning situation known as vaginal thrush. Vaginal thrush is a fungal infection caused by an organism known as Candida albicans. Females who suffer from this experience dreadful and uncomfortable symptoms such as burning, discharge, and excruciating pain that can feel like a real fire blazing. These symptoms can hinder a woman's daily life. Let's discuss how this fire is currently being put out, and then I will tell you my plan, which is my research that will not only extinguish the fire, but prevent it from reigniting. Every vaginal wall is a house with good bacteria acting as fire prevention devices. Despite protecting the house, once the fungal fire Candida burns, it burns rapidly and prevents these devices from acting out. Therefore, they are consumed and fail in their rapid race to extinguish the flames. This tenacious fire is currently being put out by buckets of water, and this is the current treatment options. This method only puts out pockets of fire, allowing the fire to spread to the rest of the house. If this is continuously allowed, the house will continuously burn. My plan is to dispatch exceptionally well-trained firefighters with the advanced fire extinguishing technology. This state-of-the-art technology will release my formulation that will not only extinguish the fire almost instantly, but coat the house in a non-flammable material, preventing any future fires. My formulation will be released directly to the vaginal wall, releasing a combination of a drug specific to killing candida and good bacteria that will repopulate the vaginal wall. This will allow for the development of a healthy vaginal canal free from infection. So let's not overlook this common infection like the common cold. We may not say goodbye to COVID-19 anytime soon, but ladies, we can soon say goodbye to the burning caused by vaginal thrush. Thank you.
All right. Thank you very much, Zakia.
Thank you.
Last off, Natasha, shall we start with you?
Zakia, we meet again.
Hi.
I believe I gave you feedback twice in the Wits round, so I'm afraid you're stuck with me again. Nice to see you at this stage of the competition. And congratulations, you deserve to be here. I think that the energy around your delivery and the passion that you have for the work that you're doing is exactly what needs to underlie good science communication. Because otherwise it all feels like something is happening in a lab and that there aren't actually people involved in doing something that will help me as a person and obviously other women all over the world. I'm interested in the kind of science of drug deployment, because I know that's obviously what you're trying to do, is to sort of deploy this drug specifically to fight vaginal thrush. I mean, there's been quite a lot of development around how drugs are deployed in the past decade or two. So I'm interested to know what got you thinking around particular forms of drug deployment in terms of vaginal thrush, kind of what research influenced your thinking and is influencing your research?
Okay, so basically it all begins with looking at current treatments and the faults that they face. So for example, you use a cream. A cream leaks out of the vaginal site. And if you take drugs orally, it will cause systemic side effects, but over the time, our bodies will develop resistance. Now, the whole thing about probiotics being used is not relatively new, but it's coming onto the market more. And currently, there's only probiotics for oral consumption to treat vaginal thrush. Now, when we take these oral and probiotics, they get eaten by the gut before anything even reaches our vaginal canal. So my whole thinking was, why not combine this probiotic and the drug and deploy directly to the vaginal canal? So this is my formulation. It's like a little pessary that has both antifungal and probiotics. So when released into the vagina, it will release both bacteria and drug. So this will overcome all the limitations. And what's great about it is that, as you can see, I'm holding it in my hand, but it's not melting. This will only melt in the presence of vaginal fluid and therefore release the drug, which will kill the vaginal candida, and then we'll have the probiotic release, which will populate the environment and then allow for a healthy vaginal canal. So if we don't repopulate with probiotics and we just treat with drug, we kill the drug, but then where's the repopulation of the probiotic? So therefore resistance develop, and then we have constant reinfection. So that's what was my thinking of developing something to not only treat, but act as a co-prevention system.
Cool, fantastic. Thank you very much. Good luck for the rest.
Thank you.
Hi, Zakia. A job well done for giving such an engaging talk. One that a lot of women resonate with. I loved your fiery opening. I loved the use of props. Some of the painting that you used, I wish I could have myself after this competition. The question that I have for you this afternoon is that you've already explained to us the ways in which the drug can be administered. But I would like to follow it up with a question to you of, are there any misconceptions that are out there as it pertains to vaginal thrush and ways in which we can treat that you wish to discredit or tell us a bit more, provide more clarity on?
So can you just repeat your question one more time? I'm sorry.
No, my question is that with so many available methods out there, yes, creams for treating vaginal thrush, you get a lot of misconceptions and that are also put out there. So are there any particular ones that you know of that you wish to discredit or talk on this afternoon?
I wouldn't really discredit anything that is out there, but I will say that when you mentioned current treatments, so as I've previously mentioned, that these current treatments have limitations. Now, I wish to overcome these limitations and to make my formulation better suited for treating vaginal thrush. Because as you know, like many females that suffer from it. So I'm a pharmacist, and when I work at the pharmacy, just one day, the amount of patients that I get that come in that want vaginal thrush medication is a lot. So although we have this treatment, they're not as effective because of the limitations. So my treatment is going to overcome these limitations. And I think also the misconception comes in as well is where some like females that do have the infection sometimes feel that they can leave it and not treat it, but they don't realize that every time it just goes away and it keeps on reoccurring. So with my formulation, by taking it, we can prevent it from returning. Thank you.
Thank you so much.
All right. Thank you very much, Zakia. That'll be all for now. Thank you for your time.
Thank you. Have a lovely day.
Bye.
Bye.
Have a good afternoon.
Hello, everyone. It is my pleasure to welcome you to the closing ceremony of the 2020 FameLab National Final. My name is Susana Galvan, and I am the Country Director at the British Council in South Africa. For those of you who are not familiar with FameLab, FameLab is an initiative of the Cheltenham Science Festivals in the UK, and it is delivered in over 30 countries through a partnership with the British Council. The British Council holds the license to deliver FameLab globally. FameLab South Africa is actually implemented through a partnership between the British Council, the South African Agency for Science and Technology Advancement, SASTA, and Jive Media Africa. FameLab has been running in South Africa since 2013, so this is actually the 8th edition of the national final, but it is actually a very unique one this year because it's the very first national final that we are delivering virtually due to COVID-19. When the lockdown started in South Africa at the end of March, we were very nervous about it, like everybody else. And actually, we saw the lockdown as a huge challenge because we are used to bringing all the participants together face to face from all over the country, from all over South Africa for a few days. But actually, I'm so glad that the team and our partners they've been able to adapt to the current challenge and the circumstances, and they've been the best use of technology to make it happen digitally, and they've also been able to attract a wider international audience for the first time. So there's a lot of positive at a time of crisis as well. So thank you so much to all the following people who have been involved in the journey to this 2020 FameLab National Final. First of all, to our partners, our strategic partners, the National Research Foundation, SASTA and Jive Media Africa. To our higher education partners, the universities and research organizations that have supported FameLab this year by hosting a total of 10 regional training sessions and heats. To all the judges that have participated in the semi-final and will be participating in the final, making tough decisions to select the winners. Thank you for offering your time at a very, very busy time for everyone, and also a very difficult time for many people. And thank you very much also for sharing your expertise with us. And of course, last but not least, to all the participants of FameLab. Of course, there would be no FameLab, and we wouldn't have had FameLab for so many years without our fantastic and very talented participants. So thank you so much for participating and also taking up the challenge of joining this year in a digital format. Finally, I would like to close this video message by saying that although we are required to select one winner who will represent South Africa at the international finals, all FameLab participants are, of course, champions already. FameLab is always associated with competition, which is expected. But in South Africa, over and above the competition element, our model has developed specific aspects that reflect local priorities. It centers, number one, on encouraging diversity, with a stronger emphasis on capacity building than the actual competition. Our approach is less about finding the best speaker and more about identifying those with potential to enrich society's understanding of various complex issues. FameLab South Africa is therefore a means of identifying and nurturing more diverse voices in the science community. With that said, I would like to congratulate all the FameLab participants for being part of this transformative program this year, and of course to all past participants for making FameLab such a successful program. And of course to all the audience out there, I do hope that you enjoyed listening to some of the very inspiring and fantastic talks by the researchers that took place in the program. I hope that you felt as inspired, empowered, and hopeful for the future as I did. So with that said, thank you again, and hope you enjoy the final. Thank you.
SASTA, a business unit of the National Research Foundation, is excited and happy to be part of the FameLab initiative, and we've been working with the FameLab for quite a while, and this is actually a strategic initiative for us because it's at the core of our business, which is to lead and coordinate the discourse on science with and for society in support of the national imperative developing a scientifically literate society. My brief today is to give a vote of thanks, and I would like to thank the following people and organizations for their contribution to the success of the FameLab initiative in South Africa in 2019 and 2020. The heats commenced in August 2019 and ended in March 2020, just before the national lockdown, which was proclaimed by the President of the Republic of South Africa, Cyril Ramaphosa. First and foremost, I want to take this opportunity to thank all the participants in the FameLab competition for this year. This year was this year we're faced with unexpected challenges that have affected the way in which we interact with each other, and in particular in this case the FameLab initiative. We'd like to commend this year's masterclass participants and hit winners in accepting the challenge and bringing their enthusiasm and passion for the science and communication in communicating the science to the virtual space of the masterclass, semifinals and finals. They are to be commended on raising to the challenge and fully engaging in this new way of working due to COVID-19 epidemic. I would like, first, to thank our major partners, because we are not naive or conscious of the fact that we will not be able, to do this and pull, this off without, our major partners, in particular the British Council, with Chunai Gozong and Meekness Lunga for their support and for linking us with, the FameLab project. I also want to take this opportunity to thank Jive Media Africa, in particular Robert Ingels, Ghani and Mazubane, who were very instrumental in all the successful implementation of the project. And then I will also want to take this opportunity to thank all the institutions and the organizations that under the circumstances decided not to abandon us, but to get involved in the FameLab project. And in particular, our line department, which is the Department of Science and Innovation, the University of Free State, the Central University of Technology, the Council for Scientific and Industrial Research and Nanotechnology Innovation Center, the Nanotechnology Public Engagement Program, the University of Venda, the University of Pretoria, the University of KwaZulu-Natal, the African Health Institute, the University of Limpopo, the University of Witwatersrand, the Tshwane University of Technology, the University of South Africa, Rose University, Nelson Mandela University, Walter Sisulu University, and one of our facilities, that is the South African Institute for Aquatic Biodiversity. And without, and I just thank, these, organizations for joining us. But at the same time, to run an organization of this magnitude, we also need, judges to assist us to determine who the winners will be. And then I want to take, this opportunity to thank and commend, the following judges for making time during these difficult, times to act as our judges. First, I want to thank Natasha Joseph from Conversation Africa for being our judge. And also, secondly, I also want to thank Nozipo Gumbi from UNISA, who I recall that she was one of our participants in the FameLab some years ago, and she ended up representing South Africa in the UK. And this is a good story to tell, because we're keeping the momentum, and our previous participants are also coming back to the program, and they are also assisting us to take this program to the next level. Last but not least, I want to take this opportunity to also thank Professor Knox Makunga from the University of Stellenbosch for also making time and joining us in ensuring that we do have judges for this initiative that we value so much. Lastly, I also want to take this opportunity to thank the SASTA team, all those people who were involved in ensuring that SASTA deliver on this big initiative FameLab.
All right, thank you very much, judges, once again for joining us through this process. Perhaps before we wrap things up, I'll just like to get a few comments and feedback from your side. Let's start off with you, Natasha.
Just to say, it's always such a pleasure to be involved with FameLab, and the work that's being done around science communication, I think, is remarkable. And to watch these energetic, brilliant young scientists who are so determined not only to do great science, but to make sure that ordinary people understand what that science is and what it entails. I was struck by the depth of the work that's being done across a variety of fields. It's an amazing kind of experience to feel like you walk away from a few hours of judging, having learned new things about trees, mosquitoes, tuberculosis, nanomedicine, a little bit of everything. And I think that the quality of science communication that's coming out of young South African scientists is something that we should all be extremely excited about.
All righty, thank you very much. Shall we then move over to Nozipho Kumbi?
Yes, thank you so much, Star. I share the very same sentiments as Natasha. It was such a pleasure to be part of this session, to listen to all the different talks. There was just so much to learn, and it's such a pleasure also to see the talents that is in the young scientists that we have in South Africa, scientists that talk to the problems and are addressing the problems that the country is facing, and problems that are not only unique to us, but problems that also resonate with other parts of the countries, and with their technologies, their research that they are doing, other countries can get benefits off from them. So that was such a pleasure to see, and also to commend you guys for the training that you've given to these young communicators. It's such impeccable to see the delivery, the manner in which they deliver some of the content that is very sensitive, but deliver it in such a tasteful manner, well thought of, well executed. It just shows how much effort all of you guys have put in, Jive Media, British Council, SASTA, everything that you've done to help, to groom, to shape all of these young communicators. It's such a pleasure to see. And just in closing from my side, I know I speak from experience. FameLab is called a competition. It's FameLab competition. And ultimately for all of us, when you hear the word competition, you immediately want to win. It's a competition, so I have to win. And so what I would encourage to the ones that do not make top 3 is that even though you do not win or walk away with the title itself, what a true win from FameLab is that you have mastered the art of delivering a tough scientific concept in such an easily digestible manner for everyone to be able to consume it. So you've done excellent in delivering and engaging others to learn from it. You might not be the title holder, but you've done a great job in delivering the science in a manner that we can all understand. So good luck, and don't stop sharing and communicating science. Thank you.
Absolutely, Nozipho. And I think definitely if you want to see the footprints of the competition in the South African context, you can look at examples like yourselves. So thank you very much for that. Prof. Nakunga, shall we close it off with you?
Yes, indeed. Thank you very much. On behalf of all three judges, I'd like to thank you for inviting us to participate in witnessing the talent that South Africa actually has. And having been around on Friday at the semi-finals, I can see incredible improvements, and today was just magic. I don't want to, speak more, because I think Natasha and Nozipho have already covered just about everything that I would have liked to have said. What I would just like to express again is how incredible the talent that we have, not only in terms of science communication, but in the science itself. It is not only relevant to our country, it's globally relevant, and it is at the cutting edge. And so congratulations to everybody that's been involved with this FameLab initiative of 2020 under extremely different and difficult circumstances. Thanks, everybody.